For REACH-MD, this is audio abstracts, produced in collaboration with the Crohn's and Colitis Foundation. I'm Dr. Ivy Law, an assistant project scientist in the Center for Inflammatory Bowel Diseases at the University of California, Los Angeles, as well as a member of the Crohn's and Colitis Foundation's Rising Educators, Academics, and Clinicians Helping IBD Group, or REACH-IBD. Today, I'll be reviewing an article published in the Inflammatory Bowel Disease Journal titled A Specific Mutation in Muc2 Determines Early Dysbiosis in Colitis-Prone Winnie mice. Inflammatory bowel diseases, or IBD for short, describe a group of gastrointestinal disorders that include Crohn's disease and ulcerative colitis. Interactions between environmental factors, host genetics, and gut microbiome play an important role in developing IBD pathophysiology. Focusing on host genetics, the authors of this article studied the prospective changes in fecal microbiota under the influence of Muc2 mutation, which is a gene encoding for mucin. Mucins are major protein components in the intestinal mucus layer. In fact, the intestinal mucus layer is both a habitat of intestinal microbiome and a barrier that separates the microbiota from the intestinal epithelial cells. To investigate the changes in fecal microbiota, Winnie mice were the transgenic mouse model used in this study because they carry a missense mutation in Muc2 and have been shown to have a less compact inner mucus layer that leads to a spontaneous development of ulcerative colitis symptoms. Winnie mice were transgenic mouse model used in this study because they carry a missense mutation in Muc2 and have been shown to have a less compact inner mucus layer that leads to spontaneous development of ulcerative colitis-like symptoms. In this study, Winnie mice and their wild-type littermates were separated by genotype and were observed from 4 to 16 weeks. During this period, Winnie mice gained less weight and had more watery stools when compared to their wild-type counterparts. Histological evaluation reviewed the development of colonic inflammation, indicated by increased epithelial erosion and immune cell infiltration in colons. After confirming the induction of colitis at 16 weeks, DNA was extracted from fecal matter collected at 4, 8, and 16 weeks and was sequenced to review differences in fecal microbiota between wild-type and Winnie mice. An analysis of 16S rRNA metagenomics showed that there were differences in relative abundance in bacterial phyla present in fecal samples taken from wild-type and Winnie mice. Furthermore, the relative abundance of genus level changed over the period of experiments and also between wild-type and Winnie mice. For example, the bacterial genus Bacteroides was in high abundance in Winnie mice than in wild-type mice over time, while the genus Paraprevotella, its relative abundance was reduced in wild-type mice over time, but increased in Winnie mice. It's also important to note that, although Bacteroides was in high abundance in Winnie mice, previous studies showed that the relative abundance of Bacteroides was significantly lower in patients with Crohn's disease or ulcerative colitis, especially in the active phase of the disease. This observation reflects the potential differences between mouse models and our disease population in pathophysiology and microbiota composition. Despite the above limitations, the results of this study demonstrate that host genetics is a driving force to modify intestinal microbiome footprint, which is an important element in IBD pathophysiology. Furthermore, this study also provides evidence showing that mutations in Muc2 or other alterations in intestinal mucus layers impact the maintenance of intestinal homeostasis and IBD pathophysiology. If you are interested in this topic or others on Crohn's disease and ulcerative colitis, the Crohn's and Colitis Foundation's Inflammatory Bowel Diseases Journal provides the most impactful and cutting-edge clinical topics and research findings. For more information on the foundation, please visit crohnsandcolitisfoundations.org. This has been a presentation of audio abstracts. To revisit this episode and others from this series, visit reachmd.com/audio abstracts. This is REACH-MD, be part of the knowledge.